So we will be recording this talk and it will be posted on our seminar page on the HMSC website. So check it out there. A couple of announcements before we get started. We will not be having seminar next week due to the short week with the 4th of July holiday. So join us for our next seminar on July 10th when Daniel Morgan, our former Hatfield artist in residence and lead innovation designer for Nike, will talk about the intersection of design and marine science, where he will share details about his 8-month residency here at Hatfield doing research and working with Dr. Chris Langdon. Now, the reason we're all here today. Our today's speaker is Angelina Zuelow. She is a PhD candidate in the Henkel Lab at Hatfield. She earned her Bachelor of Science at Humboldt State and a Master of Science at CSU Fullerton. She was also an REU intern at Hatfield in 2017 and has returned to pursue a PhD in the same lab. Today, she will present research highlights from each phase of her academic career so far. Take it away.
Okay, is my mic working? Okay, let's get into it. So today I'm just going to take you quickly through my academic journey. And basically, like Mariana said, I did my undergrad at Humboldt State University, and then I also did an REU here at Hatfield during that time. And then I did my master's at CSU Fullerton, but I like to say I also got an honorary degree from UW because most of my fieldwork was done at University of Washington for my master's. And then I came back here for my PhD here at OSU. So for my undergrad, I went to Humboldt State, which is now Cal Poly Humboldt. It had recently had a name change, but it was Humboldt when I was there. And I was there from 2014 to 2018. And I majored in biology with an emphasis in marine biology. And I worked in 2 distinct labs. I worked in Paul Bordeaux's lab and Brian Tissot's lab. And then I was also a TA assistant in invertebrate zoology. And all of my research while I was at Humboldt State was really focused on sea stars. So like I said, I was there from 2014 to 2018. 2014 through about 2016 is when we had major sea star wasting. It was extremely problematic those years. Like we were losing all of our sea stars like entirely at some sites. So basically all of the research effort at that time was to like track sea star wasting, see what's happening with the sea stars and like how the community was impacted by that. So I spent a lot of time looking at diseased stars. You can see by the upper right picture, that's a star that's pretty heavily diseased and lost 2 of its stars, or 2 of its arms. So I spent a lot of time tracking those. But then on a happier note, we also looked at their morphology, specifically their armor, and I'm going to focus on that today for this part of the talk. So the main sea star that we studied was Pisaster ocraceus, commonly called the ochre sea star, very common on the west coast of the continental US, and you can see by these photos that they really can dominate some places. These photos are from Bob Creek, which is just south of here, and they can really just like overtake whole spaces, and they are considered keystone species because they do have a pretty large impact on the communities around them. And so what I mean by armor, when we like focused on that, was their spines. So you can see by these different photos that the spines can vary pretty substantially based on the star that you're looking at, even though all these sea stars are the same species. So in some stars, they're commonly known for having this like star pattern in the middle. But some of them can have very minimal spines, which are made out of calcium carbonate, or they can have really big spines that basically cover their entire body. And they can also, like this one, the star pattern is like almost, you can't even tell that it's there, and its spines are almost in rows of 3, so really thick in terms of how many spines it has on its body. So we wanted to know, how does this spine density or armor vary, and does it vary by habitat? And then also, how do the spines vary, if they do at all? So what I mean by habitat, we looked at 4 very distinct habitats around Northern California. First, we had our 2 wave exposed, so we had Boulder and Bench, and they get a lot of wave exposure. You can see Boulder in the upper left, classic Boulder field, very rocky, and then that bottom left photo is like a bench site. This is Kibasilla Hill in Mendocino County, and just like very long, flat, going out, not a lot of boulders, but both very wave exposed. And then on the right are 2 protected examples. So we use the bay, and this is Humboldt Bay, which is still very high salinity, so very similar to the open ocean, but flat. Like that water is flat, the jetties are flat, but you get a lot of sea stars there. And then we also had a reference site, very protected because it was the aquarium, and this was Point Defiance Zoo and Aquarium, which is up in Tacoma. And we used a bunch of stars from inside of those tanks that had been living in those tanks for decades. So they were kind of like our reference stars. And then this is a map of just where our sites are located. So this is, again, Northern California. And you can see we had 2 boulder sites, 2 bench sites, 2 bay sites. And then, of course, Point Defiance Zoo is up in Washington, so not depicted here. And then just to give you reference on where this is in relation to Humboldt State. Humboldt's kind of in the middle there in Arcata. And then the Marine Lab is up just past Baker Beach up in Trinidad, California. And so what did we do with these stars? Essentially, we took hundreds of stars, we photographed them, and then we processed these data through ImageJ. And basically how we used ImageJ is that you can take a single grab, right, of the star, and then you can tell ImageJ to only focus on the central disk, and then you can invert the colors, and it will actually count the spines for you, which is very much faster than having to count the spines yourself. So it gives you a final spine density number, because it's also calculating the area of that central disk. So that's how we got the final numbers. And then this is what the spines look like under SEM. So we actually dissected a bunch of spines off of these Pisaster. You can just remove them really softly with forceps. And then you basically have to bleach the spines to get rid of all the tissue that's stuck around them. And then you can put them through a scanning electron microscopy, and you can see these kind of photos. So this is just an example of what one spine could look like off of the top of these sea stars. And so first I'm going to show you alberoll spine density, which is again how dense their spines are on their surface. And then on the x-axis here is radius. And what I mean by radius is just the length of the longest arm to the center of the disk. And so main takeaways here, again, I've color-coded all the habitats. But main thing I want you to take away is that there's a negative relationship between these two things, essentially meaning the bigger you get, the less dense your spines are going to be, which makes sense. As you grow bigger, your spines are just going to be kind of spread out. And then that negative relationship was only significant for the boulder and the bay sites, so not the other two. And then I do want you to focus up here where you see only boulder. And so these are really small stars. These are little baby stars, and their spine densities are through the roof. So the juveniles tend to be very armored, very covered in spines, and specifically really at the boulder sites. You're seeing that really dense spines. And then here's some more photos of what the spines look like. So they can vary pretty substantially, which of course we didn't know this until we processed these through SEM. And so we were able to actually name these different types of spines in the paper that we just published, which is very exciting. But basically you can see that their base can vary pretty substantially. So this blunt example has a short base, and then overall kind of just straight top. And then you have a columnar one where it's really straight, and the base itself is really long compared to this other type. And then we have tapered, so they're also really long, but they start to crown at the top. And then we also had convex. I like to call this one the mushroom shape because it's got a short base and then really rounded at the top. And then these other two are a little more zoomed in, so you can see the pores, and also just the crazy difference in how it's growing out of the top, which is really interesting. So we named all of these, and then we just took as many spines as possible from as many stars from a bunch of different habitats to see if the ratio of these spine types varied by habitat. And surprisingly, it really does vary by habitat. So what I've got here on the y-axis is proportion of spine types, and then the habitats are on the x. And we've put the most common spine type above each of the habitats. So you can see for the bay, it was mostly that tapered shape spine that we were seeing. That's 50% of the spines that we pulled were that tapered shape. And then for the bench and boulder sites, it was almost entirely that convex or that mushroom shape. And then for the reference site, which again is the one where they've been living in those tall aquariums for a really long time, all of their spines are just straight up, growing straight out of their body. So pretty interesting to see. And then to connect this to wave exposure, because that's what we think is driving this, we also put out Claude cards, which is a kind of a proxy for wave exposure. So we put a bunch of those out, and you can see that the two wave exposed sites were definitely the most wave exposed sites. You're seeing a lot of Claude card disappearance, which means there's a lot of waves moving or water moving through there. So the bench and boulder sites definitely have more wave exposure. And then for the sediment, the bottom graph, that's just we put out these traps that will catch sediment if it's available in the water. And basically the only sites that had a bunch of sediment were boulder. So this might lead to the idea that the dominant spine types that we're seeing are really driven by wave exposure and scouring from sediment, because we saw like that mushroom shape almost entirely in the sites that have the most wave exposure and the most sediment. So to conclude here for this section, I want you to remember that the highest spine densities were associated with more wave exposed habitats, and spine types do really vary by habitat, which is really interesting. And of course, the mushroom shaped was the most common one that we saw, and we really do think that it's like naturally they can just grow straight up, but they're being pushed down by wave exposure and scour from sediment. So, of course, while I was at Humboldt, I also did an REU, and I came here to OSU. It was my first time at OSU, and I worked in Sarah Henkel's lab with shrimps. And as I said, everything before now has been sea stars. So this is my first time working with shrimps. And now I'm going to bring in marine heat waves. So if you're unfamiliar, marine heat waves are described as anomalous high sea surface temperatures that are sustained for at least 5 days. And we had the Blob here in 2014 to 2016, which really enveloped the Northeast Pacific, and it really remained in some places for up to 2 years, which is a crazy long time for something like this to happen. And this picture on the left shows you like how extreme that anomaly was in some places. This picture is from May 2015. And the colors are like the dark red is about 3 degrees Celsius high, that's like an extreme movement. And you can see that a lot of the Northeast Pacific was heating up. And so here's a quick timeline from NOAA Fisheries that shows kind of how this developed and then where it went away. Basically 2013, we were doing okay. We still had upwelling in the Northeast Pacific. But then in 2014, we had wind die down and the blob started to develop. We also had low snowpack for the 2014-2015 winter. And then 2015 is when the blob was like at its peak. That's when temperatures were really elevated, and we also had an El Nino develop. So it was like a double whammy in 2015. And then 2016, waters were still pretty hot in Alaska. So you can see by that picture, that Alaska, the Gulf of Alaska, is still really red, but in terms of like us over here in the Pacific Northwest, we were doing okay. And then by 2017, relatively everything had cooled except some like outer open ocean areas. So doing okay. And then I'll just remind you that I was here for my REU in 2017, so I missed all of these years, and I was really oblivious to all of this happening because I was an undergrad, and I was really focused on sea star wasting, and at the time, we had no idea what was causing sea star wasting. We just knew it was happening, and it was something that we were monitoring. So I was oblivious to the blob going on, which is fine, because luckily, Sarah had been collecting for many years before, during, and after this whole period. So we had a lot of data to work with. And so the star for this section is Crangon alaskensis, which is a common benthic shrimp in Oregon. And basically the collections for this study were done from 2010 to 2018 and in different phases. So for 2010 to 2015, they did a bunch of trawling. It was about 3 to 5 times per year, so like almost every season. 2016, there was no data collection done because it was like in between funding sources. And then it picked back up by NOAA in 2017 and 2018, and that was done 6 times per year. So we have a lot of samples to work with. And then on the right is a map of what the sites look like. So you can see we're down here in Aquina Bay. Hatfield is right here. And so these are just 3 like main transects where the boats were going out and trawling to collect for these samples. And so this is what it looks like for how like going out there. Most of this was done off of the Alaka here at OSU, but some other boats were used too. And then with these samples, they always put a CTD out, which takes a bunch of environmental data, such as depth, dissolved oxygen, conductivity, temperature, all that stuff. And then on the right here is what the beam trawl looks like. It's 2 meters across, and it gets pushed off of the back of this boat, and then it's trawled on the bottom for 5 to 10 minutes, depending on which month it is. And so, of course, Crangon is not the only thing you pull up off of a beam trawl. So all of this has to be sorted first. And so you sort everything, you pull out the shrimps, you sort them by species, which gets really complicated. And then you freeze them. And then they were frozen for many years. And then I came here for my REU and was like, I want to do something with all of these shrimp. So that's what I did. And I put them through a bomb calorimeter. And if you've never worked with a bomb calorimeter, that's okay. I hadn't until I came here. And what it looks like is that bottom left photo, which is not very exciting. But the bottom right photo kind of shows you what's going on inside of the bomb. And how this works is you have to have a dried sample, some type of dried organic material, and it goes into this chamber right here, and then this white piece is considered the bomb because it gets pressurized to 30 atm, so pretty volatile if you dropped it. And then it gets put into a water bath, and then basically you can ignite this sample from the outside. So the sample will burn inside of this chamber, it will release energy into the water, and the water temperature will go up. And so from that, you can back calculate how much energy is coming out of these samples. And so I just want to highlight how the REU is different from what I did for my undergrad at Humboldt State. This was my first time working physically in a lab. So all of that C-star stuff I described, that was field-based. We were doing all of that in the field and then processing and sitting at the computer. Like none of that was really in the lab. And also for this REU, I did no field work. I was not on the boat collecting these samples. Like these were samples that were collected for me, which is good because I get really sick on boats. So I'm thankful to everybody that collected these samples for me. And this was also my first time like doing a full proposal and report, which I had not done at HSU. And so now I'm going to show you some of the highlights that I found while I was an intern, and then I will circle back to this project in a little bit. So first, here's some data from, so these are the samples that I was able to process from 2010 to 2015. On the y-axis is joules per gram, so this is just caloric density, or how many calories are in the samples. And then on the x-axis is the months, and then the years are color-coded. And what I want you to focus on here is that basically from April to August, it's always increasing for most of the years, and with a peak in August, so just like summer highs for their caloric density, which is really common for a lot of our species here. And then you just see decreases from there on. So fall and winter are pretty low productive months in Oregon in general. But then if you look at 2015, which is this orange heat wave year, it's the first year where we saw a really steep decline from June to August. So definitely seeing a huge decline there. And then if you look a little more closely at these differences, this is just showing you the difference in the caloric density from June to August. So you can see that basically before the heat wave really hit Oregon, it was always positive, always increasing into that summer, whereas in 2015, we see a huge decline. So pretty interesting results. I was able to present this stuff at SACNAS, which was my first time ever at SACNAS. SACNAS stands for the Society for the Advancement of Chicanos and Native Americans in Science. I had never heard about this until Yi-Chung asked me why I wasn't going. So I'm really thankful that I learned about this organization. I've basically been working with them since then. So great organization. And then I was also able to present at CERF, which is the Coastal and Estuarine Research Federation. Yay, Yi-Chung. And like I said, I'm going to circle back to these because I was only able to finish 5 years of data, but we had more years of data, and we got somebody else to come back and do it for us, which is amazing. So after I finished my REU, I went back to Humboldt to finish my bachelor's, and then I also did 2 back-to-back phycology courses at the end of my senior year. I took phycology with the late Frank Shaughnessy from Humboldt State. That was a great course. That was my first time ever being introduced to algae. I had not taken a course on them prior to this. And then I also went to University of Washington, Friday Harbor Labs, and took one of their summer marine botany courses. And that's taught by Tom Mumford and Wilson Freshwater, who are very enthusiastic people about algae, which is really important. Because basically this is where I learned that like, oh, I'm an algae person. I want to look at algae for the rest of my life, which is a good thing to learn about yourself. So I went on to grad school. I did my master's at CSU Fullerton, which I will just put a note here that I decided on CSU Fullerton because Sarah Henkel, my REU advisor, also went to CSU Fullerton, and she had a really good time there. We had different advisors, obviously, because she's a decade ahead of me. But really good recommendation on that university. And that's kind of really good way to learn how to decide where to go if you're thinking about grad school, is to just ask people about their experiences, where they've been, who were their advisors, that kind of thing. So I ended up working with Jen Burniford, who did her PhD here at OSU. And I was there from 2018 to 2021. And I switched my focus from sea stars and shrimp, and I started working on kelps. So I worked with Egregia and pictured on the left. And I was really focused on the presence and the effects of them on the communities around them. And I did duplicate studies, so I had a site in northern Washington, and I had a site in Southern California. So I was basically at the 2 edges of its very distinct geographic range. But I'm not going to focus on this because I want to skip ahead to the REU and really stay on marine heat waves. But if you want to go and read my 150-page master's thesis, please do. It took me forever to write. So I'm going to skip ahead and come now to grad school here back at OSU. I arrived back at OSU in 2021. So now I'm a PhD student in integrative biology, and I'm also back in Sarah's lab, which I'm very grateful that she let me come back to OSU because I love it here. And my PhD is still focused on kelps, but I'm going to come back to the Crangon shrimp story because a year into my PhD, we got a new REU intern in 2022, Hope Donahue, and she came from the University of New Hampshire, and she came in and she processed the samples that I never got to in 2017. So she came back, she processed all the 2017, all the 2018 years. So now we have a complete data set of 8 years of Crangon. So I can show you what that looks like when we add 2017 and 2018. So now I'm showing you still caloric density on the y-axis, x-axis instead now is day of year, and it's broken up by the months. And so you can still see that the orange here is 2015. So still 2015 was the lowest year in August. And now we've added 2017, which is this like X labels here. And you can see that 2017 was in like basically following in the middle. So the caloric densities were kind of average throughout the year, but it did have a really high peak in September. And then even crazier, 2018 is this one that has the asterisks as the symbols and had the highest August of any August we had seen so far. And it also continued to increase into the fall and December, fall and winter months, which is really surprising for this data. So now when we look at the August anomaly, so this is just zooming in on the summer, you can see that it's calculating an average here across the years and then how every year deviates from that. And so basically every year before the heat waves was basically always being more productive in August, higher caloric densities. But then when you get into 2014 and 2015, you see a decrease in those caloric densities, with 2015 being the lowest. And then if you look at 2018, it's crazy high up here and definitely had like the highest year yet for 2018 for August, indicating really intense recovery for this species. And then we wanted to know, okay, well now we know caloric densities are impacted by the heat wave, but what about the heat wave is impacting them? And so we took all of the CTD data, so like all the environmental data, and then we also looked at oceanographic indices. So this could be the upwelling index or the beauty and cutie, which are another type of upwelling index. And then we also looked at climate indices, such as the PDO, which is the Pacific Decadal Oscillation, or the ENSO, which is the El Nino Southern Oscillation. And we ran a ton of models to find which data was most explaining this caloric density trend we were seeing. And so the final model, which ended with the highest R-squared and the lowest AIC, had beauty as the upwelling index, specifically with a 1-month delay, which is really important. And then the PDO for the climate index. So we just used one. And then this is the final model to explain how we were seeing the predicted caloric densities. And basically, temperature, salinity, fluorescence, and dissolved oxygen were all included because they made the model stronger, but it was only temperature and dissolved oxygen that were significantly impacting the caloric densities. And then we also had beauty in there. Like I said, we had a 1-month delay. So what you can do with the beauty, you can pull all this data off of NOAA still right now, which is good. But basically, it will give you a monthly number for what the beauty is or the upwelling index is for that month. So you can either take that data, you can take the month before, or we did 2 months before. So we checked all 3 of those to see which ones had higher predicting power, and it was always a month before. Which makes sense because if you change nitrates like in the water, you're not going to see an immediate change in the caloric density of these shrimp. It's going to have a little bit of a delay related to like what their diet is. And so this is what the caloric density looks like against the beauty index. Beauty does stand for biological effective upwelling transport index, but beauty is a funner name to say. And like I said, it is calculated with a nitrate flux focus. So really focused on how the nitrates are changing in the water. And you can see that the years are color-coded, but overall this is a really strong positive relationship with the 1-month delay, and the simple linear model showed really significant relationship. So higher beauty, higher caloric density. So to conclude, finally, the 8 years of data for this REU, like I said, the Blob did have a significant reduction on the caloric densities of Crangon, especially in 2014, and really strong in 2015. And then 2017, which was hypothetically 2 years post-Blob, caloric densities didn't fully recover to that pre-Blob value yet. But then in 2018, we had the highest caloric values of all the years, which indicates recovery is definitely possible after a big event like this. Especially with enough time and the right conditions. And then, like I said, the beauty had the strongest significant positive relationship with caloric densities. And then I was able to present this final work at Western Society of Naturalists, which is another really great marine biology conference and just like group in general. They have a conference every year. The next one coming up is in San Diego, so maybe think about it. And this is my first time giving an oral presentation at a conference because the ones before had all been during COVID and on Zoom, which is kind of like not as scary. And then we were able to finally publish this work in 2024. So I did this work in 2017. We had another REU come back in 2022. Finally put it into a paper in 2024. Yes. Okay, so now I'm going to transition a little bit to, we're still at OSU now, but now I'm going to focus on what I'm doing for my PhD. And this is just a quick infographic to show you how things are still heating up for us. We might not be in marine heat waves right now, but we're definitely seeing warming of the ocean. So you can see in the Northeast Pacific, we're definitely red, we're getting hot. And then this is mean sea surface temperature for the globe. And you can see that basically since 2013, sea surface temperature is just going up. We're not seeing any dips down below this average. So we're getting hotter every year. And this is unfortunately leading to a lot of kelp forests disappearing, especially here on the West Coast. So this is a perfect example. This is the same exact site by these two photos. And you can see the bull kelp is nice and happy on the left and then fully gone on the right. And this is understory here and some like differences between what's happening on the rocks underneath all these canopies. And you can see that urchins are starting to take over here and we're like losing a lot of our other types of algae underneath. And this is really being connected by temperature and overgrazing. And specifically, overgrazing is really being driven by the loss of predatory sea stars, which was connected to that sea star wasting event that we had over a decade ago that is still plaguing us today. So this is an example of thermal tolerance for bull kelp. So this is the bull kelp up here, and this study basically tracked their growth, and then they exposed them to a bunch of different temperatures. And what's important to note here is that for the bull kelp, it really maxes at about 12 or 13 degrees Celsius. But once you start to get above that, it really starts to decline in its ability to grow and survive. So this kind of shows how if we enter these kind of heat wave periods or El Niños begin, the temperatures get too hot, bull kelp is just going to start to decline pretty heavily just from its physiological tolerance. And then this is an example of what it looked like pre-blob, and then they also were able to do during blob and post-blob. And this is for Mendocino County and Sonoma County, which are Northern California counties. And the green pictured here is all the kelp cover. 2018, it was nice, or 2008, it was nice and lush and covering every county, and things were doing well. 2014, the heat wave began. You could see some, but definitely not to the extent. And then by 2015, it was like basically all gone. And then post-blob, some returned in Mendocino County, Sonoma County fully devoid of these kelps and these kelp canopies. So I'm going to focus today on a couple canopy formers. The first one I want to talk about is Egregia menziesii, which is a feather boa kelp, is commonly called the feather boa kelp. This one dominates the intertidal zone, but it is also found subtidally. It's a perennial, so it can live for many years, which is different from bull kelp. And it does have a really large geographic range. It's found all the way from Alaska down to Baja California, which is pretty distinct because a lot of kelps tend to have like half of this range available to them. And so here at OSU, specifically at Hatfield, I've been running separate lab experiments. I did 5 just with this species alone, and I did them between 2022 and 2024. And I exposed them to a bunch of different temperatures depending on the experiment. So I did 10 degrees Celsius up to 18 degrees Celsius. And just to give you reference, Oregon sea surface temperatures are between 10 and 11 degrees Celsius. So I was definitely upping what I was exposing them to. And then basically I grew them in these tanks in the experimental seawater facility for many weeks and tracked their photosynthesis, their respiration rates, their growth, and their caloric density. So I'll just show you one of the experiments here first. This is gross photosynthesis on the Y. So this is like how much their photosynthetic rate is, accounting for respiration. And then on the x-axis is days. So this experiment ran for 24 days, and I exposed them to one of three temperatures, either 13 degrees, 14, or 15 degrees Celsius. And what I want you to see is that on days 0, 6, and 12, there's no significant differences between the treatments, but they do look like they're starting to separate out. And then by day 18 and day 24, the highest photosynthetic rates were correlated with the highest temperatures. So they were actually photosynthesizing more at higher temps. And for the caloric density, I'll remind you, I learned this skill in my REU to learn how to use a bomb, but I came back for my PhD and was like, what else can I use the bomb for? I'm going to bomb as many kelps as I possibly can. So that's what I did. So I took all of the kelps after this experiment and dried them and put them through the bomb and found that, again, the highest caloric densities were associated with the highest temps. So the ones that were maintained at these really high temperatures had higher caloric densities compared to the other two. But if you go above 15 degrees Celsius, the kelps do not have a good time. So this is an example of what happens to them. On the left, you can see that this kelp has fully disintegrated, so you can't even tell that it's a kelp anymore. I know that it is because that's what it was when I put it in. But it's changed since then. And then on the right here, you can see the tissue almost looks like it's melting on itself. Definitely not doing well at 15 degrees Celsius. So for this experiment, again, I'm showing you gross photosynthetic rate on the Y, and on the X, this one ran for 28 days. But here I exposed them to 14, 16, and 18 degrees Celsius. So definitely got hotter for these treatments. And on day 14, I had lost almost half of the individuals for this experiment, mostly out of the 14 and 18 degree group. But what's really interesting is that once I lost them here, the ones that survived the rest of the time and had no tissue loss. So that means like the two individuals that were left over in 16 and the two individuals that were left over in 18, no effect of temperature to them. And they also had higher photosynthetic rates. So those ones were totally able to survive, which is really interesting to see. And now I will introduce the final canopy former I'm going to talk about today, which is Phyllospadix scholari. And this is a surf grass, and it is an angiosperm, which means it's a flowering plant. It's like one of the only categories of true like flowering plants that we get in the intertidal. And it does also dominate in really large beds like you can see on that photo on the right. And it also has a very similar range to Egregia. So they occupy very similar spaces. And these are some photos of where they overlap. So for instance, this is Fogarty Creek, which is just north of here. And basically all the green is Phyllospadix and all the darker patches are Egregia. So like if you skipped through the intertidal, you'd be bouncing between these two canopies basically throughout this entire site. And then on the right, this is just a tide pool where you can see both are occurring together again, Phyllospadix here with Egregia on the right. So I saw this a lot, and there's a lot of long-term data that shows that they can change dominance depending on what year it is. And so I was really interested in kind of figuring out who is better suited for a warming ocean between these two species. And so for the final experiments, I've been doing a field experiment where I'm tracking them in the field, but today I'm just going to be talking about the lab stuff. And basically I did the same stuff that I just did with Egregia that I just talked about in the lab, but I added Phyllospadix also. So now I have two species together at the same time. And so for the first experiment, this one ran for 28 days, and I exposed them to either 13 degrees Celsius or 15 degrees Celsius. And the y-axis here is gross photosynthesis, and the x-axis is day. So this one ran for 28 days, and then the two species are separated out. And what I really want you to focus on is that Egregia is doing the same thing that it's been doing, which is good. Replication is good to show that at higher temps, it does photosynthesize more. And I did see that in this experiment. And these asterisks mean significant differences between the treatments on that day. So on days 7, 14, and 21, it was photosynthesizing higher in higher temps. But Phyllospadix, I saw no significant differences across any of the days, but you can see that there's huge variation in this species already. So even on day 0, so day 0 represents before they went into these temperature treatments, and you can see the variation is already pretty large in what this species is doing photosynthetically. And then as you introduce the temperature treatment, the variation gets way bigger in the ones that were heated to 15 degrees Celsius, but still not significantly different from what's happening in the ambient treatment. And so after doing that, I was like, okay, well, that doesn't really tell me which of the species is going to do better in a crazy marine heat wave. So I was like, how far can I push these species? So for my experiment 2, this one only ran for 7 days, but the ambient started at 15 and 16 and like hovered between those 2 degrees. And then the heated treatment, every day I added a degree of temperature. So that means by day 7, they were maxing at 23 degrees Celsius, which is 13 degrees higher than what the ocean like is at right now. So these should be extreme temperatures for these species, okay? So now I'm showing you, instead of putting it on the left, I'm doing metabolic rate, and then I've separated the photosynthetic rate from the respiration rate. And then I've got the 2 species, Egregia is on the top, Phyllospadix is on the right, or on the bottom. And what I want you to take away from this is that for Egregia, you can see that it always had a higher photosynthetic rate in the heated treatment until day 5, when it hit about 21 degrees Celsius. And then you can see that very distinct flip where the heated treatment is now photosynthesizing less, and then they separate even more out as you go further down in temperature. So not huge significant differences between these, but there is an overall trend that the photosynthetic rate does start to go down once you hit 20 degrees Celsius. But if you look at Phyllospadix is flat. No significant differences between any of these days, between the ambient and the heated trials, even though that on day 7 here, this is hitting 23 degrees Celsius. Phyllospadix is just cruising along. And then for their respiration rates, which is just this represents what they're respiring just in the dark. And you can see that respiration continued to go up, and we had some extreme individuals getting really stressed out and respiring really high. And these were significant basically across the board for Egregia, but only 2 days were significantly different for Phyllospadix. So respiration increased in both of the species, but Phyllospadix, it was less extreme and definitely seemed to just like cruise along for them. So study conclusions here is that Egregia definitely has or is a very robust kelp to warming conditions, but it does have a lower upper thermal limit compared to Phyllospadix. So if we keep increasing ocean warming, which is definitely going to happen, it might lead to surf grass kind of dominating over kelps around here. So in conclusion, I have talked about 3 very distinct projects that I've had a lot of fun learning through. And the main highlights I want you to take away is that for sea stars, specifically for Pisaster, the spine density and type really varied by habitat, which is pretty interesting to see. For Crangon, again, another example where like something is suffering because of these marine heat waves. The caloric densities of Crangon were really negatively impacted by the heat wave when it was happening. And then for Egregia, for one of our really common intertidal kelps, it does have a high thermal tolerance, which is kind of good news for the next couple of years of warming. But if we start to see marine heat waves exceed 20 degrees Celsius, then we're going to start getting in trouble with our kelps in general. And then I'll highlight, of course, all of the funding that went through all of these and all of my amazing mentors that put me through all of this. Humboldt State, Paul Bordeaux, and Angela, amazing. Loved working with them. And we just put out our new paper, so definitely go look at it. And then for my REU and OSU, of course, Sarah, who I'm still working with today, and Ichan, yay. And then I would love it if you check out my website because all of our publications are listed there. And then I'll take any questions now. Yay. Hold on.
Wonderful job. Thank you. May we all be as unbothered as Phyllospadix under changing conditions. Online, go ahead and drop your questions in the chat, and we'll go ahead and start taking some questions in the room. Any questions for Angelina? All right, Icha.
Love that slide of the REU one with OSU with the Bewley model. You had a paragraph.
Let's see.
A great presentation, by the way. Amazing work. So wonderful to see all this amazing journey that you've gone through. One more, I think. Nope, keep going. Keep going.
This
One more. There.
Okay.
That last paragraph, the final model with the highest R2 and lowest AIC included beauty with 1 month delay. Could you elaborate a little bit more on that?
Yeah, so basically we ran a bunch of different models, and each one included one climate indice and one like upwelling indice. So we were trying to see which like explained the most, if that makes sense. So then the final one, like the one that always had the highest R squared and lowest AIC, which AIC stands for, I had to put it on my notes, Akakite's information criterion, which is like a way to balance goodness of fit and complexity. So like we'll tell you like your model's too complex or there's too many parameters in this model, so then the AIC will fluctuate. So you want the lowest AIC. So like we were trying to hit like which model is the best for that, right? And it ended up being the beauty as the like upwelling index and the PDO as the climate index. So that means like the other upwelling index could have been cutie, or it could have been just the general upwelling index, and both of those were like less significant. And then for the climate index, it could have been either PDO or ENSO, and PDO was always stronger. So.
And can you elaborate a little bit more on the significance of that 1 month delay?
Yeah, so I think I was surprised to see it was 1 month. I thought maybe it would be the 2 month delay would be stronger, because I pulled all the data, right, and then I correlated it to either like if we took the samples in January, I correlated it to January beauty, the December prior, or the November prior. And it makes sense that it wouldn't be the exact month, because like similar to if you decide one day, like I'm going to stop eating sugar today, you're not going to see a huge difference in your body the next day, right? You're going to see like a delay in that metabolism and how it changes. So it makes sense that it wasn't the exact, but I was surprised that it was 1 month and not 2 months. So just the 1 month seems to be like a stronger correlation than the 2 months prior to when we took the samples.
Great, thank you. Wonderful presentation. Thank you.
All right, any questions online? No, questions in the room?
What could be the potential impacts of the seagrass taking over the kelp, and like how would that affect the other ecosystems?
Oh, great question. And I have taken a bunch of data on that, but I have not looked at the data yet. But basically in the field, what I've been doing is we set out a bunch of transects 2 years ago, and they were either Egregia, Phyllospadix, or they were joint plots. And then we either like destroyed them, so ripped out everything, or we sheared it. So it was like the canopy disappeared, like from an imaginary heat wave event or a rip out or something like that. And then we've been tracking the community. And in general, I mean, diversity is lower in surfgrass beds. So like we know that in general, but I don't know if that is like, if the diversity lower is necessarily a negative on productivity. So definitely need to look at the data more and see how the plots have switched over the last 2 years. We just sampled a couple days ago for the final plot, so I need to look at the data now. But I would imagine that, yeah, if surfgrass takes over, we're gonna see species disappear, right? That are used to kelp cover, but we might not see like loss of diversity in the intertidal in general. So, but definitely surfgrass beds get real sandy, so they actually will like tack sand to their rhizomes. So we'll shift the environment pretty substantially, I think.
Questions? All right.
First I wanted to say thank you so much for spending time to talk to us today. And then I wanted to say, with ocean acidification and the spines of the starfish being made of calcium carbonate, was there anything that you discovered with those maybe affecting like the shape of the spine or anything?
Yeah, so for that morphology work, of course, we only chose asymptomatic stars. So they were the ones that were not exhibiting any signs of wasting, because that would have totally skewed our results. But of course, like when they go through wasting, they are like their body is melting, right? So that involves the calcium carbonate too. So I would think that yes, if they're going through strong wasting, their spines will also break down. And then in terms of ocean acidification, of course, yes, they use calcium carbonate to make their body. But I don't know if anybody's found heavy correlations between like OA and C star production. I know it's like most of the work has been studied with clams and like bivalves and things like that, that I think rely on it a little more to create their outer structure, whereas sea stars still have connective tissue and like, you know, all of this other stuff on their arboreal structure. But I would imagine, yes, as OA gets worse, right, they won't be able to make their calcium structures too. So
Thank you.
not great.
Great talk, by the way. I wanted to ask, based on the sea stars that you surveyed, you saw some very distinct different shapes in their spines. Is it possible that in other ecosystems that you didn't have a chance in with different like wave action, that there could be even other types of spines that you like didn't take a look or didn't discover yet?
Yeah, definitely. And a really interesting thing that I know Angela, who she's the master student I worked with, now she's doing a PhD at Northeastern, and she wants to look at the spines of like stars that are in the Caribbean because they look totally different, right? And the structures and the crown of thorns, like the one that grows, sure, what's going on there? Why are they doing that? You know? So of course she's really interested in that stuff. I like it too, but I'm a kelp girly now. But yeah, I mean, this is just one example, right, of where like we figure something out and now we're like, okay, now we have to go look at it in everybody else and see like, how does this look? Because of course these were also only done in Northern California. So like, are they different here in Oregon? I don't know. They probably are.
Awesome, thank you.
Yeah.
Other questions? Way up there, making me work for it. Okay, I'm coming.
Sorry. I was wondering, could you elaborate more on the trends that you observed between the spines and the habitats for the different Pisaster species, individuals? So like, the difference between individuals at the aquarium or sites of high wave intensity and like what causes those differences perhaps?
Yeah, so I think, let's see if I can pull it up. But basically, the ones that were in the aquarium are just to give you understanding of how long those stars, like these are full adult stars that have been in that aquarium for, I think it was like over 20 years when we sampled them. So they were like resident Pisaster that have just been living the good, calm life in the aquarium. And then all the other stars that we took were like straight out of the field, right? And we used those like different habitats, and those were those three. So like bay, bench, and boulder are like the three habitat types that we have in the intertidal. And yeah, so we were just looking at the differences between those, and definitely like, of course, the aquarium has no wave exposure. Like even if you're talking about like the pumps that siphon in water, like Pisaster doesn't care about that. They're just like, oh, this is a great happy day for me. Whereas the ones at bench and boulder are like getting really intense wave exposure constantly. And we also saw that right with the Claude cards, which are like big, like heavy plaster cards you could put out there that will disintegrate over time. And the faster they disintegrate, it means they're like the wave exposure is really intense there. And yeah. Anything else
And
further I can elaborate on?
if you go back to the slide with the different shapes of the spines, what might be the benefit to having some of those specific spine shapes?
Yeah, so of course, the one thing we didn't do, and it is really hard, we did try to do this and it did not work out. First, I'll talk about Pisaster is genius. They can get out of anything that they want to. So if you try to tag them to try to track the same star over time, they'll just like drop an arm because they're like, no. And if you try to like color code them, we tried that for a while. We were like dyeing different arms to try to track the same individual in time. They also can like flush their tissue out. So very hard to track stars. But one of the things, of course, that we want to do is like from this only tells us, right, that that's the spine of each, like that's dominating each habitat. But we don't know if they started as something else and became that over time, which is what we think is happening. We think that like if you're in a perfect environment on the reference side, right, those spines are going straight up and that's just, they just grow straight out of your body. And then on the left, they're being worn down by the environmental parameters. But we can't say that, right? That's what we think is going on. And so the next thing, right, would be to like transplant these stars and then look at their spines over time, right? But really hard to transplant Pisaster. They are too smart. But we did try. They didn't like it.
Thank you. Great presentation.
Thanks.
All right, coming down there. Anything online, Jamie?
Yeah, we've got one question in the chat. Joe says, thank you for a great talk. And they're wondering, for kelps that have experienced tissue damage from higher temperatures in your experimental trials, do you think they would recover if given enough time at a lower temperature?
So in one of the experiments, we did do that. Last year, the REU, 2024 REU, Martin, he did that. So we did like 7 days of marine heat wave exposure, and then we did 7 days of like recovery. So it went from like 18 degrees Celsius, dropped back down to 10, and they did not recover. So it could have also not been enough time. Like we only gave them 7 days to recover, and then we processed them for caloric density. So maybe longer time would have allowed them to do that. But something to think about too is like if they start to fully disintegrate, like it's really hard to come back from that. And one of the things that I think is going on is that it's not necessarily like the kelp itself self-destructing. It could also be the microbial community on top of them. And so like if that's fully shifting, then it's going to shift like their ability to do everything that they normally do. And some recent studies have come out because there's a lot of work now on microbial activity, which is kind of what I want to get into, but I'm also not a microbiologist, but that have found like the microbial community on kelps is totally different from what's in the water, and it will shift with temperature. So that's probably what's I would predict is what's happening with these kelps.
I'm so sorry we keep asking questions about the sea stars,
Yeah,
but
that's okay.
now that I'm
This paper just came out. I'm really excited about it.
okay, good. Now that we're talking about it, I was curious, based on the previous question, if there's any like with the spine differences, if there's anybody looking into like possibly like PCR differences with like genetics, if there's within the populations, or if it's like specifically environmental pressures or combination.
Yeah, like is the mushroom shape just how that's growing, and it's
Yeah.
like a genetic thing? I have no
Right.
idea. I know some people are doing this, like Angela is doing it for her PhD, but she's doing it on Asterias, which is kind of like the East Coast version of our Pisaster, but they don't get as big and they don't have as many spines. So, Pisaster is just a sexy species. But yeah, I don't think this paper just came out, so hopefully it will like inspire other people to spend their money to look at this. I don't have money to do PCR. I wish I did. I would also love to look at the spines like in other places too, especially in like the Puget Sound. Like, do they look totally different there? And other species. There's another Pisaster giganteus, which has huge spines. What do they look like?
Right. Yeah. Exactly. Weird.
Hi.
Hi.
I had 2 questions.
Okay.
So for the tagging or following a sea star, would something like the tagging in otoliths in fish work similarly? 'Cause it's the
Yeah, so
same-
otoliths you're talking about when they come out of the fish ears and they have rings on them.
Yeah.
So Pisaster, I mean, you can't even age a Pisaster because their growth rate depends on like how much they're eating and what they're eating. So sometimes you'll see a Pisaster and be like, oh, that looks like a baby, and it's like been sitting in an aquarium for 20 years, and you're like, oh, okay, that's much older than I thought it was. Pisaster can live for a really long time too, so it's really hard to gauge how old they are. And then they don't have like growth patterns because they can drop arms, right, and regrow an arm. So then it's really hard to gauge like how old they are.
But would it work? Because I believe for some of them, if you like, they kind of it's a glow in the dark. Maybe I'm misremembering, but you can just see that it's the same star by a certain glow that they have.
Yeah,
and
you
can
can.
look.
Yeah, you can tag them. Yeah, totally.
So would it work, you think?
I think it does. It's so like the way we did it, because for like a spine to change, that's not going to happen like within a tide, right? It's going to
Oh,
happen like
sure.
over a long period of time, especially if you're moving them between habitats. So we were like red dyeing their arms, and we would do different arms on the spine. So it's like if we did arm 1 and 5, we knew what star it was. But then first of all, finding them like a month later is like tragic because they can move so fast. And then if we did find them, they can like shift their connective tissue. So it would be either really faded, or we would be like, I think this is 1 in 5. I think this is who it's supposed to be. But yeah, we would only get like 30% return rates, which is really low. You want to be able to like track as many as possible. So we couldn't like determine any conclusions from that.
And then a
But yeah,
second
glow in the
question.
dark is interesting. I wonder if
Oh,
you
yeah.
could like write names on them.
Maybe
Name each one.
make a little pattern with the little circles.
Yeah, I'm always jealous of people that work with bivalves because you can just like marker on the outside of a bivalve and it's like, yeah, it's the same one. Can't do that with Pisaster. They're too fleshy.
And a second quick question is, why did you decide to do the comparison between seagrass and kelp when, I think no wait, this could sound wrong, but, why did you decide not to compare like the giant kelp, bull kelp, and the feather with boa kelp?
Yeah, great question. So here in Oregon, we do get the bull kelp in the intertidal, but it doesn't survive to adulthood. It will just like settle, it will grow a little baby, and then it will eventually get like out crowded by the other kelps. And then macrocystis, which is the giant kelp, we also have here in Oregon, but it's only offshore. Like very distinct populations, kind of hard to find, but then you'll find it like on the beaches, right? And you'll be like, where are you coming from? I know you're coming from somewhere, but not intertidal. So Egregia is like the dominant intertidal kelp. The other one that we have here is Heatophyllum, which is the like sea cabbage. And I did some work with sea cabbage, but they produce different canopies. So the sea cabbage is more upright, whereas Egregia is like long canopy forming. So I was trying to look at that comparison of like the function that it's producing. But definitely like Egregia and Heatophyllum can be in the same place also at the same time. And the inspiration for working with surfgrass is not because I love surfgrass, because I love kelp, but it's because there's some PSCO data, which is like PSCO is one of our long-term monitoring groups on the West Coast. And in Oregon, there's a really interesting like data set that they have where like literally they tracked Egregia and Phyllospadix for like 20 years. They do it at all the sites. And a few of the sites, they are switching dominance between the two. And it doesn't, I like took all the data and I mapped it against El Niños, and it doesn't quite match with temperature, but it's like a year off for some. And so I wanted to know like, why are they switching dominance? And is it only temperature? Is it something else? So that's kind of what I'm trying to get at. So that was the inspiration for surfgrass.
Thank you.
Yeah. Definitely look at long-term data sets. Like there are interesting things in long-term data sets that we do not look at. So I was like, what can I do with this? This is crazy.
Awesome. Great questions, everybody. I'm going to hold you for one more second. I know we're right at time, but I'm going to ask a selfish question here. We have a lot of young budding scientists in the room right now, REU students, interns, all of that. So I wanted to know what your biggest takeaway was from your REU experience. This could be personal, it could be scientific, it could be anything in between there. What would you say your biggest thing you learned in your REU was, and how we can inspire the future?
That's a tough one. That is a tough one. Biggest thing I learned from my REU. This is a life lesson too, but say yes to more things. So for example, when I was applying to REUs, I knew I wanted to work with Sarah because she had a really interesting lab, but you know, the project is already kind of determined for you, depending on if you're coming in with funding. And she was like, I have a shrimp project, and that's all I have. And I was just like, okay. I guess I'll do that. So I said yes to that. I said yes to, you know, going to my first conferences related to that data. And then I said yes to joining committees after that. And I would say, yeah, biggest takeaway is that say yes to more things if you're interested in them. And it's okay to not love your project. It's okay to not want to work with what you're working with at the end of your REU. I don't work with shrimp anymore, but I loved my REU experience. So that's what I would say.
All right, well, let's give her one more round of applause. Thank you, Angelina. And thank you to those that joined us online. You are all free to go. If you didn't already, please sign in so I can tally all of you for cookies and coffee for the future. But otherwise, thank you. We will see you not next week, but the following week on July 10th for Daniel Morgan's talk. Thank you.